Even when arranged into higher levels of organization, such as tissues and organs, cells are an organism's basic unit of structure and function. Overall, evolution is the unifying biological theme, but we need to realize that all cells are related by their descent from earlier cells, but have been modified in various ways during the history of life on Earth. For example, the picture here provided is a picture of a blood sample. We see red blood cells, we see white blood cells, we see platelets. All of them are derived from the same cell line, but they are very different in structure and function. So diversity accumulates over time. The three major contributors to the cell theory, Schleiden, Schwann, and Virchow, overall, they acknowledge within the cell theory that the cell is the fundamental unit of structure and function of life, and that all organisms are made of one or more cells, and that those cells arise from other cells through cellular division. That basic idea has been expanded upon over time to incorporate the idea that cells carry genetic material that they pass on to their daughter cells, but that all cells are essentially the same in their chemical composition and their usage of energy. So the basic contributing idea here is cell theory, as laid out by Schleiden, Schwann, and Virchow. To actually study the cell, you need to first look at microscopy. Before we go into microscopy, though, you need to make sure you understand the difference between magnification, resolution, and contrast. Magnification is the ratio of an object's image to its real size. So a light microscope, for example, can magnify effectively to about 1,000 times the real size of a specimen. But for those kind of things, you're limited to the wavelength of the light that you are using. So we'll get there more in detail later. Resolution is a measure of image clarity. It's the minimum distance two points can be separated and still be distinguished. Okay, whereas contrast is looking at the differences in parts of the sample, which can be improved by using different types of staining, for example, or labeling. You can see some examples here of light microscopes with various contrasts. So light microscopy is the older form of microscopy. It's been discovered since the early 1500s. In a light microscope, visible light passes through the specimen and then through glass lenses to allow that focus. The lenses refract light so that the image is magnified into the eye or camera. It can resolve a fair number of things. We can see light microscopes on the range here, and as we've used higher and higher energy levels of light, we've been able to see smaller and smaller things going into that super resolution microscopy. But you're limited to about half the wavelength of the light. So a higher energy light, like lasers, for example, can see smaller things than a lower energy visible light spectrum. If you need to see something smaller, you need to move to something that is a lot higher energy, which would be electron microscopy. So for smaller structures, you're going to use beams of electrons through which a specimen can be seen. Resolution is inversely related to wavelength, so we can see much smaller things because there's a much shorter wavelength. Theoretically, we can improve this to get incredibly small, and some of the modern ones can see down to individual atoms. There are many different types of electron microscopy, but the main two that you're expected to know are the scanning electron microscopes and the transmission electron microscopes. If you're trying to see sort of the exterior, the scanning electron microscopy is sufficient. If you need to see the internal microstructure, then you're going to have to slice it and use transmission electron microscopy. Either way, though, nothing living can be done within electron microscopy. It's too high energy. So if you need to view a living organism, you're going to have to use a light microscope. If you want to use an electron microscope, you are going to have to kill the organism, and you are going to be unable to view dynamic processes. But electron microscopes keep advancing. You can see here, for example, a carbon structure with the bonds actually being visible within the electron microscopy, so it can definitely see incredibly small. Now, if you actually want to study the parts of the cell, then you're going to need to isolate the cellular components. To do that, the most typical way is to suspend the cells within a fluid and then to break the cells, to disrupt the cell membranes through some form of lysis. You then homogenize them, which basically rupture the cell membrane and blend them together so that everything is free-floating and not contained by membrane. After you have done that, and it could be with something like sound or detergent or whatever, you're then going to fractionate, which means you're going to spin at increasingly higher levels in a centrifuge. Okay, so for example, the heaviest things are going to settle first, so I'm going to spin at a low speed, and then the whole cells, the ones that didn't break, and any nuclei and things, the large components will settle to the bottom because they weigh the most. I'll pull all the fluid with all of that suspended supernatant and spin it again at a higher speed. That separates out increasingly smaller and smaller and smaller things until all that you're left with is the smallest components like ribosomes and viruses. Once you've done that, once you've isolated the sections by size, you can then examine the properties of those components. What are the actual limits to how big a cell can get, though? Eukaryotic cells are generally larger than prokaryotic cells, but the logistics of carrying out metabolism sets limits on cell size. The smallest bacteria, the mycoplasmas, are about 0.1 micrometer. Most bacteria are between 1 and 5 micrometer. Eukaryotic cells between 10 and 100 micrometer. Unfertilized eggs would be the largest cell. So I guess that would be something like an ostrich egg, for example. But as cells increase in size, its volume increases faster than its surface area. Why? Well, the surface area to volume ratio is a square-cube relationship. Surface area increases on the square, volume increases cubically. So it's proportional to a linear dimension squared for area, whereas volume is proportional to a linear dimension cubed. So if you take the surface area and divide it by the volume, you get the surface to volume area ratio. And a larger surface area to volume ratio is more likely to function. It's going to be able to metabolize, it's going to be able to dispose of waste, it's going to be able to take in raw materials. Without that relationship, that is a problem. There is another limit to cell size, the so-called G to V ratio, or the genome to volume ratio. Basically, a given amount of genome can produce a certain amount of instructions, and a larger cell still only has the same amount of DNA, so only so many instructions can be produced. So there are some other limits here as well. What can you do to maximize surface area other than becoming massive cells? Well, you become more cells, like we saw in the last example, or you alter your shape and structure, flattening, branching, folding, projections. These are all major ways that cells increase their surface area without altering their overall volume. So we understand, I hope, that as things get bigger, their insides, their volume gets more bigger than their outside surface area. It's not a random relationship here. It's a major function of living cells. If you actually look at a prokaryote, prokaryotic cells are pretty simple. They come in a couple of basic shapes, rods, spheres, spirals, but their actual structures are fairly limited. All cells are enclosed in a plasma membrane, a selective barrier made of usually some form of phospholipid. Inside the cell, you're going to have cytosol, okay, and if you have the cytosol and the suspended materials, we then call it cytoplasm. All cells contain chromosomes that carry their DNA. All cells have ribosomes, which are tiny complexes that make proteins. But prokaryotic cells have a couple of differences. They do not have any membrane-bound organelles, so they do have organelles, like we said, like ribosomes, but they don't have anything that's membrane-bound. Their DNA, their chromosomes, are found in the nucleoid, typically as a single large circular chromosome. They have a cell wall, and some of them can have capsules and flagellas and those kind of things that we can see on the outside here, but those are the major differences. Within the cytoplasm of the eukaryotic cells, there's a variety of membrane-bound organelles, each with specialized form and function. These membrane-bound organelles are absent in prokaryotes. Actually, the outer membrane of a bacterial cell is most of the membrane that it has. For eukaryotic cells, the outer membrane is only about 2% of the membrane. The rest is internal in all these organelles. It still has ribosomes and chromosomes and cytoplasm, cytosol, just like that, but it has other things as well. We'll get more detail later on. Overall, you can see here that plant and animal cells have many similar structures. These images here are sort of idealized cells. You have way more of these organelles than are shown, and these are indicative only of the most common organelles. But you should see overall that the centrosome is a structure here that is in animal cells. That is unique to animal cells. We'll get into why later. And in plants, you have a cell wall, just like in bacteria, even though it's made of a different sugar. You have plasmodesmata, that's openings in the cell wall. Since we don't have a cell wall, we can't have a plasmodesmata, so those are going to be interlinked. And then you're also going to have that chloroplast, which is a representative of a larger group of organelles called the plastids. We do not have those chloroplasts. That's pretty much it for the basic cell structures, the major differences between plant and animal cells. There are a few other differences that we'll get into as we go further, but that should be a good starting place for now.